Hello students, today we have topics about enzymes. Let's start with definition. The definition of enzymes is that they are biological catalysts responsible for speed up the biochemical reactions. About the nature, there are two types. Mostly they are globular proteins, no fibrous, but globular proteins. And some of them are RNA, or the other name is ribozymes. But from today we'll talk about only proteins, not ribozymes. The biological significance of enzymes is that they can be tested in the blood or in some other biological metrics like saliva, and we can say assessment, and we can understand about the pharmacological regulation of enzymes, and because they are key elements in diagnosis and therapy. The similarities and differences with other catalysts. The enzymes are used for the increase both the forward and the reverse reactions. They do not change their equilibrium. They elevate the rate of only spontaneously occurring reactions by providing alternative route with lower activation energy. And this is the most important feature for enzymes, because they decrease the activation energy of the reaction. Only small amount is necessary, and they remain unchanged in the end of the reaction, which means they can be used again and again in the next reaction. The differences are that they are highly effective, highly specific, they act in mild physiological condition, and the most important feature of all enzymes is their regulation. We have to know their regulation, because if we know how they can be regulated, we can modify their activity outside by pharmacological control. This graph shows the progression of the reaction with or without enzyme. If the reaction is without enzyme, the barrier is high and the reaction is slow. If the reaction is with enzyme, the barrier is low and the reaction is fast. Enzymes lower activation energy. They accelerate the reaction without shifting the equilibrium. Why is possible to decrease the activation energy of the reaction too much? Because of the formation of some intermediate complexes, which are called enzyme substrate complex and enzyme product complex. Thanks to these complexes, there is possibility to rapidly providing alternative reaction pathway of the enzyme. Enzyme nomenclature, how the enzyme name is formed. Actually exists some ways to form the enzyme name. The first one is according to so-called enzyme nomenclature, which is international unit about the formation of enzyme names, which is according to some individual number and according to the classification of enzyme reactions. According to this classification, exists a specific code number of enzymes with 4 numbers. The first number shows the main group of enzyme, the second number shows the subgroup, subgroup, and individual number. But in practice, we can use this formation of enzyme name, which is according to first, substrate name. We need to know the substrate name of the reaction. Number 2, the type of the reaction, plus A's in the end. Let's see the examples. Malate dehydrogenase means that the malate is substrate name. Dehydrogenation is a type of the reaction, plus A's, which means, let's see the second example, phenylalanine hydroxylase. What is the substrate name? Yes? Phenylalanine. Phenylalanine. What is the type of the reaction? Hydroxylation. The next example, pyruvate carboxylase. Substrate name? Pyruvate. Type of the reaction? Carboxylation, yes. Some exceptions. After the product name, this example is citrate synthase, which means that citrate is not a substrate. This is the product of the reaction. And the name is formed because of the product. We need to know that there is difference between synthase and synthetase, but we'll talk about that in the end of the presentation. Another example is a lactate dehydrogenase. Look about this example. We have this pyruvate, this is the substrate, to lactate. This is the product of the reaction. Substrate, product. The reaction is reversible, and the enzyme name is lactate dehydrogenase because of the reverse reaction, not because of the forward reaction. Understand? This is the exception. One more after the source. Salivary amylase. Salivary amylase means that the place of action of this enzyme is in the mouth. Pancreatic amylase. Pancreatic amylase means that not the place of action, but the place of synthesis of this enzyme is pancreas. Where is the place of action is another question. The place of action is small intestines, but the place of synthesis is pancreas. That's why the name is pancreatic amylase. Lingual lipase, in the mouth again. After the type of regulation, hormone-sensitive lipase. This means that this enzyme is active because of some contact with different hormones. Some common names. Pepsin. Where is pepsin? In the stomach. Trypsin. Small intestines. Amylase. You understand about amylase either in the mouth or in the small intestines. Structure of enzymes. Students, actually there are two types of enzymes. Some of them are only proteins, with only protein part. These enzymes are simple ones. Only protein part they have. But some of the enzymes are combination between protein part and non-protein parts. These enzymes are complex enzymes or coenzymes. The protein part is called apoenzymes. The non-protein part is called cofactor. There are two types of cofactors. According to the bond. According to the bond. If the bond is covalent or tightly bound to apoenzyme, this cofactor is called prosthetic group. If the bond is non-covalent, which means loosely bound to apoenzyme, the cofactor is called coenzyme. One question. Tell me why we need to eat everyday vitamins. Why we need to eat vitamins? Yes? Not exactly. Actually, the answer is because they are or they will be converted to cofactors of the enzymes. That's why. They are or they will be converted to cofactors. Which compounds can serve as cofactors of vitamins and their derivatives? Nucleotides. There is complex cofactors composed of combination of vitamins and nucleotides, and metal ions. Metal ions can also serve as cofactors. Actually, they are prosthetic groups because of the covalent bonds, the contact with the protein part. For example, zinc is used as a cofactor of carboanhydrase enzyme or carboxypeptidase. Magnesium is used for all kinases, hexokinase, glucokinase, phosphofructokinase, and etc. Heme, this compound is called heme, there is iron in the center. This molecule is used as a prosthetic group of cytochromes. These are compounds that are composed from heme plus proteins, these are called cytochromes, as a prosthetic group of some enzymes like catalase, heme peroxidase, endothelial nitric oxide synthase. And we'll start talking about water-soluble vitamins first, but before that, I'd like to discuss something about the differences between water and lipid-soluble vitamins. Students, we said last time something about the differences between water solubility of the compounds. Why we need to know if they are water or not water soluble. The first reason was because of their transport. Because you understand that the blood, the cytosol, are water-soluble solutions, and all water-soluble vitamins can be transported in them alone, independently, without specific transporters, with some exceptions, but mostly without transporters. But if we're talking about the fat-soluble vitamins, they cannot be transported alone. They need to be transported with specific transporters. All transporters are proteins. The second difference is about the resorption. Water-soluble vitamins can be absorbed, like all water-soluble compounds, directly through enterocytes to the blood, and in the blood independent transport. But when we talk about fat-soluble vitamins, students, we need to explain how can be absorbed in the small intestines. Look here. This is the lumen of small intestines, and these are the enterocytes, and these are the apical surfaces of all enterocytes, nucleus. After the intake of fat food or lipid food, here, all this food is degraded to monomers, and after that, these monomers enter the enterocytes together with fat-soluble vitamins. After that, to be absorbed in the blood, thanks to the basal membrane, this is the blood, there is possibility of the formation of specific particles which are responsible for the transport in the blood. These particles are called chylomicrons. In the chylomicrons, in the center, there is TAG, triacylglycerols, which are the main fat from the food, and all fat-soluble vitamins, which are DEKA. Plus, we said we need some transport protein, which is called ApoB48. All this particle is called lipoprotein because of the combination between lipid and protein part. And there are 5 types of lipoproteins. One of them is this one, which is called chylomicrons. To summarize, fat-soluble vitamins can be absorbed in the blood only thanks to the formation of chylomicrons, but the formation of chylomicrons require taking of fat foods, which means if you want to take some fat-soluble vitamins as supplements alone, it cannot be without fat food. For example, spoon of milk or some oil, but without fat food, you cannot absorb your fat-soluble vitamins. This is important about the resorption of fat-soluble vitamins. Something else about the toxicity there is, and about the saving there is, because lipid-soluble vitamins can be saved in our body. The organ of save is liver or adipose tissue, and that's why if you exceed the dose of some fat-soluble vitamin, it can be toxic for you. But it's absolutely opposite about water-soluble vitamins. They, because they are water-soluble, they can be released via the urine very easy out of the body, which means we need daily intake, everyday intake of water-soluble vitamin. If you today take some fruit, tomorrow you need again, because of their water solubility and very easy releasing via the urine, that way. And let's start one by one with water-soluble vitamins. You have to know the number of the vitamin, you have to know the name, the full name, which is thiamine, and you have to know the coenzyme name. It's different, sometimes it's different. You have to know the catalysis reactions, the specific examples of enzymes that use this cofactor, and if there is food deficiency, you have to know the name of this food deficiency. B1, the other name is thiamine. This is the name, thiamine, and it's only the B1, but it's not the cofactor. To become cofactor, needs to be phosphorylated twice, to form the full cofactor name, which is thiamine pyrophosphate or diphosphate, is the same. Most often is used pyrophosphate. TPP is a cofactor of B1. Which are the enzymes? This cofactor is used in transferase reactions for the transfer of C2 groups. This is in the structure of pyruvate dehydrogenase complex, alpha-ketoglutarate dehydrogenase complex, and one more enzyme which is called transcatolase from pentose phosphate pathway. The food deficiency of B1 is called beriberi. The disease is called beriberi and includes chronic peripheral neuritis and polyneuritis. The second water-soluble vitamin is called B2. The full name is riboflavin. This is the riboflavin, but like a riboflavin only is nothing. If there is no combination with one phosphate atom, second phosphate, one more pentose, and one more compound which is nitrogenous base adenine, riboflavin is nothing. After all this combination, will be formed two types of cofactors with riboflavin, and these are FAD. All this is called flavin adenine dinucleotide, which is FAD, or FMN, to this phosphate atom, this is called FMN, or flavin mononucleotide. Both of the cofactors, students, can be reduced, which means that FAD and FMN are their oxidized forms. FADH and FMNH are reduced forms. This is a pair, oxidized, reduced form. This pair, which includes the reduced and oxidized form of one compound, is called redox pair. Which are the reactions that use these cofactors? The general name of all these reactions are oxidoreductases, but you have to know specifically where exactly are they used. Not only oxidoreductases, because you understand that there are many subgroups of oxidoreductases, and actually FAD is used in the reactions of dehydrogenases, and this is the example of succinate dehydrogenase and acyl coenzyme A dehydrogenase use FAD as cofactor. Also, amino acid oxidases, xanthine oxidases, some other oxidases can use these cofactors too. But mainly is FAD. This is the most common reaction, succinate dehydrogenase, because this reaction we can find in the Krebs cycle. The deficiency is very rare, with angular stomatitis, glossitis, peripheral neuritis, or secondary pellagra, so-called. I think yes, you will see this reaction today in the succinate dehydrogenase reaction. The third water-soluble vitamin is called B3, and the other name is niacin or PP or nicotinamide. And look where is the vitamin part of this cofactor. The full cofactor name is NAD, which is nicotinamide adenine dinucleotide or nicotinamide adenine dinucleotide phosphate if instead of this X there is phosphate group. If there is phosphate group, it will be not NAD, it will be NADP. There are two possible cofactors, NAD, which can be reduced to NADH, and NADP, which can be reduced to NADPH. Again, redox pairs, again reactions in oxidoreduction or oxidoreductase reactions, but exactly about these two pairs, you need to know the direction of their using. What is the direction of their cofactor? The NAD students, this cofactor NAD, is used in the reactions of dehydrogenation, which means there is NAD enters the reaction, NADH will be produced. This reaction is called dehydrogenation. Example. I will give you one common example, like in the last time, by the way. This is the substrate, and this is the product of the reaction. NAD enters, NAD plus is called the oxidized form, and the product will be NADH. NADH plus H plus is the written part, the written name. But orally, you can say just NADH is enough, or NAD it's enough, not NAD plus. NAD is enough. This is the reaction of dehydrogenation. But when we're talking about NADP, it's another situation, because NADP is used in the opposite reactions. The opposite reactions are called reactions of, do you know the opposite name of dehydrogenation? The opposite is reduction. That's why NADP, NADPH, this redox pair is used in the direction of reduction, which means two hydrogen atoms will be involved in the substrate and product will be produced in the end. That's why, to summarize, NAD and FAD, both of them are used in the reactions of dehydrogenation, or they are cofactors of dehydrogenases. NADPH is a cofactor of reductases. These are the differences. There are many examples of dehydrogenases. Lactate dehydrogenase, this enzyme that you know, malate dehydrogenase, glutamate dehydrogenase, and many dehydrogenases, they are in the cells. The full deficiency of B3 is called pellagra. It's the disease with 3D. The first D is diarrhea, second D, dermatitis, and third D, dementia, with neurological symptoms. Why the name is dinucleotide? Why is dinucleotide? Because it's very similar to be two nucleotides if we accept this like base. Because look, this is the base, pentose and phosphate. What was the combination between base, pentose and phosphate? All this combination means nucleotide. Base, pentose, phosphate is a nucleotide. And here there is almost the same. It's not base, but it's almost like base. Pentose and phosphate, again, like two nucleotides. That's why the name is accepted like dinucleotide. B5, this vitamin, the full name is pantothenic acid. Pantothenic acid is used in two structures in our cells. The first one is the structure in coenzyme A, and the second structure is so-called acyl carrier protein in the synthesis of fatty acids. The catalyzed reactions are transferase reactions with transfer of some acyl groups. Deficiency is very rare, and you can see the differences between coenzyme A and acyl carrier protein. Look at the structure of coenzyme A, students. First, we have again one base, which base is this one? It is adenine. Yes, adenine. This is 1 pentose, but phosphorylated pentose, because on the third position there is phosphate group. Then 1 phosphate, second phosphate. Then start the pantothenic acid structure. This is the pantothenic acid, which is combination between pantothenic acid and beta-alanine. Beta-alanine, actually we don't have beta amino acids in the protein structure, our proteins. We have alpha amino acids, but that's why for us it's a vitamin. We need to take it from the food. All vitamins, we cannot synthesize the vitamins, with some exceptions you see about vitamin D, for example. In the end of the coenzyme A, there is one cysteamine. Cysteamine is a product which comes from cysteine, which is amino acid. That's why you have to know that in the end, because in the end of the compound there is sulfhydryl group, which is SH. That's why in the abbreviation of coenzyme A there is written in the end SH. You can write with or without, never mind. But you need to know why there is SH group in the end. And the second compound with pantothenic acid in our cells is called acylcarioprotein. This compound is used in the fatty acid synthesis. It's a process which requires one multi-enzyme complex, which is called fatty acid synthase complex. Let's see the differences with coenzyme A. There is a serine amino acid, serine residue, and there is contact with phosphate, 1 phosphate, then pantothenic acid, and in the end cysteamine again. It's very similar to coenzyme A. The next vitamin is called B6. There are many groups and types of B6. Pyridoxine, pyridoxal, pyridoxole, pyridoxamine, but the active form, the cofactor name is pyridoxal phosphate. Pyridoxal phosphate is this one. This is the active form of B6. Where is used pyridoxal phosphate? It is used in the reactions of transferase reactions or some ligase reactions, mostly in amino acids metabolism with the transfer of amino groups and carboxyl groups. What will be the name of the removing of amino groups? When there is removing of amino group, what will be the type of this reaction? Can you understand? It's deamination. It is called deamination. Or transamination, when there is transfer of amino group. In both of the processes participate B6, pyridoxal phosphate. That's why the example enzymes are amino acid aminotransferases like ALT, AST, in decarboxylases, all decarboxylases, and some specific examples with allosynthase or in catabolism of tryptophan. Deficiency, seborrheic dermatitis and microcytic anemia, and there is some epileptic form with deficiency of B6. B6 can be saved in small quantity, by the way, it's an exception. H, vitamin H or biotin. Can you see the role of biotin? It is used not as coenzyme but as prosthetic group. Why? Because the contact with APO enzyme is covalent bond. That's why biotin is used as a prosthetic group. And after the contact with the epsilon amino group of lysine residue in the protein part of the enzyme, there is formation of one peptide covalent bond. That's why there is a prosthetic group action of biotin. Where is used biotin? Biotin, students, is used in the carboxylation reactions, but only in lipids and carbohydrates, not in proteins. In proteins there is another cofactor which is used in the carboxylation, which is vitamin K, but not biotin. Some details about participation of biotin. These are two examples: acetyl coenzyme A carboxylase, pyruvate carboxylase. One more example: propyl coenzyme A carboxylase. you need to distinguish the carboxylation from decarboxylation. We said that in decarboxylation participates pyridoxal phosphate, but in carboxylation participates biotin. Carboxylation means that we take one carboxyl group and will be involved in the substrate. Decarboxylation means opposite, removing of carboxyl group from the substrate. One more. When there is participation of biotin, all the time, every time, there is using of energy. ATP is used and is degraded, is hydrolyzed gamma bond to ADP plus P. It's obligatory. If there is biotin, don't forget biotin plus ATP to ADP plus P. There is one protein in RAOX which can inhibit biotin resorption, which is called avidin. B9, the other name is folic acid. Folic acid has active form called tetrahydrofolate or tetrahydrofolic acid, the same. Which are the catalyzed reactions? The catalyzed reactions are transferase reactions with C1 units. C1 units can be hydroxyl methyl groups, formyl group, formimino group, and some other C1 units. The structure of folic acid consists of one pteridine ring, one paraminobenzoic acid, and glutamate residue. Exactly here on the position 5 and 10, there is contact with these C1 units. And after that, they can be formed absolutely different forms of folic acid. The double bonds between 5, 6, and 7, 8 are the sites for reduction. 5 and 10 are sites for attachment of C1 units. Where is used? In desoxy-TMP synthesis or timidylate synthesis, in the purine de novo synthesis, in the synthesis of serine and methionine, in choline synthesis. The deficiency of folic acid is very important because leads, especially in pregnant women, to neural tube defects or spina bifida and megaloblastic anemia. That's why in all pregnant women they need to be supplemented with B9, not only during the pregnancy and before that, 2-3 months before that too. Need to start this supplementation with B9. Actually, students, tetrahydrofolic acid is formed from folic acid in these two reactions, in these double-step reactions. We take folic acid as a vitamin from the food, but it can be synthesized only in microorganisms. Only in microorganisms by these two reactions. From one pteridine precursor, there is formation of paraminobenzoic acid, then formation of folic acid, and then we take it from the diet. In our organism, folic acid undergoes double-step reduction reactions thanks to dehydrofolate reductase enzymes, which are NADPH dependent, which means NADPH is a cofactor. Why? Because we said that NADPH is a cofactor of all reductases, all of them. And first, dehydrofolic acid, because 2 more hydrogen atoms there are, and after that, tetrahydrofolic acid, 2 more hydrogen atoms are involved in the structure. You can see some black lines here, here and here. The black lines means possibility of blocking of these enzymes thanks to some drugs like sulfonamides. They are used to stop this reaction but only in microorganisms because this reaction doesn't exist in the humans. And methotrexate is used to inhibit this enzyme. It is used in the treatment of childhood leukemia as an anti-cancer drug. B12, the other name is cobalamin. In the center of this pteridine ring, there is one cobalt ion, And there are different types of B12 according to X, this residue, methylcobalamin or adenosylcobalamin. Actually, students, in our organism, there are only two reactions with B12. Two reactions with the two enzymes there. And these are methylmalonyl coenzyme amidase reaction, which is part of beta oxidation of fatty acid with odd number of carbon atoms. And the second one is methionine synthase reaction, where there is regeneration of methionine from homocysteine. But you'll see these reactions in next presentations, in the next lecture, one by one. All these enzymes you'll see one by one. That's why don't worry if you cannot memorize everything from today. Yes. B12 students has many exceptions. First exception is about the resorption. It's not so easy, the resorption of B12, because there is one specific intrinsic factor or cofactor which is produced in the stomach, and after the combination between this intrinsic factor plus B12, there is formation of one complex thanks to this intrinsic factor, and after that they can be absorbed in the small intestines. That's why if we need supplementation with B12, cannot be peros, but need to be to bypass the gastrointestinal tract directly by injection in the blood, maybe intramuscular or subcutaneous. And one more exception, B12 has its own transporter in the blood, which is called transcobalamin 2, unlike all the rest water-soluble vitamins which can be transported independently. It has specific transporter. There are many consequences after the deficiency of B12. It's not only these ones, but these are the main. Pernicious anemia is the first one, or megaloblastic anemia, leukocytopenia, thrombocytopenia, neurological complications with demyelination, and the high level of one compound which is called methylmalonyl. Methylmalonate, the full name is. But you'll see when we talk about this beta oxidation of fatty acid with odd number of carbon atoms. Something else there is about B12 more, it's about this reaction. Because in this reaction here, there is participation of B12. If there is no B12, never mind that the enzyme is correct. Never mind that the enzyme is intact. No cofactor. If there is no cofactor, no enzyme reaction. And if there is a deficiency of B12 here, homocysteine will be increased. Very increasing the homocysteine will be. Why? Because it's a substrate reaction. It's atypical consequences. If there is no enzyme or no action or no cofactor, never mind, the consequences will be the same. Which means high substrate and no product. But here the substrate is homocysteine. But homocysteine is a toxic factor for the blood vessel wall. Which means if there is B12 deficiency, cardiovascular risk will be increased immediately. Vitamin C or ascorbic acid is an redox pair too, like NAD, NADH, FAD, FADH, NADP, NADPH. One more redox pair is ascorbic acid because exists ascorbic and dihydroascorbic acid without two hydrogen atoms. Ascorbic acid, students, is used as the same structure as ascorbic without conversion of other form. This is the cofactor. And it is used in many, there is actually many applications of this vitamin. The first that you know from the first lecture, by the way, it's using as cofactor of two enzymes, prolyl and lysyl hydroxylases. Do you remember the synthesis of collagen? In the synthesis of collagen, we said something about so-called post-translational modification of these two amino acids, and proline will be converted to hydroxyproline, lysyl to hydroxylysyl. These two reactions with the participation of vitamin C with molecular oxygen and both of these enzymes. This post-translational modification And these amino acids is used during the formation of collagen. If there is enough quantity of vitamin C, will be form stable collagen. That's why if there is scurvy, which means full deficiency of vitamin C, the main symptoms will be bleeding, gum bleeding, but not only from the gum, from many organs can be this bleeding. Why? Because the most location of collagen is around the blood vessels. In all blood vessel wall there is collagen. That's why if there is no stable collagen, no stable blood vessel wall will be. That's why bleeding will be the symptom. Many other applications there is. Actually, I'd like to say the participation of vitamin C in the resorption of iron, for example. Iron students are two types, 3+ from the plants and 2+, which is involved in the heme. This is called heme iron, which is in the meat. But if we take plant food with iron 3+, it must be converted to 2+ in the apical surface of the enterocytes. This conversion of 3+ iron to 2+ iron needs ascorbic acid again. That's why if you take iron as supplement, it needs to be together with vitamin C, or just one fruit together with iron. Yes, it will facilitate the absorption of iron in the blood directly. And some other applications there are in the dopamine hydroxylase reaction and some other reactions there. And that's all about water-soluble vitamins. Now some words about the lipid-soluble vitamins. Students, to memorize all fat-soluble vitamins, memorize the word DECA. If you memorize this word DECA with some comma, DEKA, you never forget any of these fat-soluble vitamins. The first one is vitamin K. It exists as 3 forms, K1, K2 and K3, and it is used as a cofactor of one enzyme which is called gamma-glutamate carboxylase. Do you remember that I said something about vitamin K today? I said something about biotin, which is a cofactor of carboxylases in lipids and carbohydrates, but in proteins it's not biotin, it is vitamin K. This is the cofactor of carboxylation in proteins. Not proteins, of course, I'm talking about amino acids. And there is only one amino acid that can be carboxylated, and it's glutamate. Glutamate can be carboxylated to gamma-carboxyglutamate. And the role of gamma-carboxyglutamate is formation of one more carboxyl group to be contacted with calcium. This is the role. To contact with one molecule of calcium. Where is used this reaction? This reaction is used in the blood coagulation factors, which are number 2, 7, 9, 10, protein S and protein C and protein S, and in the bone tissue osteocalcin, and in matrix GLA proteins, which are used as inhibitor of soft tissue calcification in the arteries. Let's see the reaction. Vitamin K is used with 3 forms. The first form is hydroquinone form of vitamin K, which enters the reaction together with molecular oxygen and carbon dioxide. After that, the second carboxyl group is gamma place, on the gamma place is here, and vitamin K turns to vitamin K epoxide. This is the second form of vitamin K. Then, vitamin K epoxide need to be restored by 2 reactions, reductase reactions, with vitamin K epoxide and reductase enzymes. If there is blocking of these enzymes by specific anticoagulants, will be formation of blood clot. That's why these drugs are called anticoagulants, or the full name is indirect anticoagulants, right, like warfarin and coumarins. They are used exactly in these processes of blocking of vitamin K epoxide reductase enzyme. Why we need to take vitamin D and vitamin K together? Because of the last using. Because students, vitamin K is used not only in the carboxylation of blood clotting factors. It is called again post-translational modification, by the way. It is used to direct the calcium from the arteries to the bones. From the arteries to the bones. Otherwise, can be a formation of tissue calcification if there is not enough quantity of vitamin K. The next vitamin is called vitamin A, which is called, there are actually three forms of vitamin A, biologically active: retinol, retinal, here is aldehyde group CHO, here there is only OH, and a retinoic acid. A retinoic acid is used as a hormone, and they are obtained from the plant precursor beta-carotene, from carotenoid family. If we're talking about plants, if we're talking about animals, the form is a retinal, this one. The vitamin A is used in the regeneration of 11-cis-retinal in the visual cycle. Let's see directly the scheme. This is the biological role of vitamin A. It is used for the formation of rhodopsin, which is the combination between opsin is one protein, plus 11-cis-retinal. 11-cis-retinal, during the light, is combined with opsin to form rhodopsin. After that, there is dissociation and division of the compound to opsin and transretinal. Then, transretinal in the dark undergoes a reduction process thanks to NADH, which is the cofactor here, and transretinal turns to transretinol. Then, isomerization of transretinol to 11-cis-retinol. Then, dehydrogenation thanks to this enzyme plus NAD as cofactor, and 11-cis-retinal is regenerated. The clinical significance is that deficiency of vitamin A leads to lack of 11-cis-retinal, this compound, impaired rhodopsin formation, and night blindness. Genetic mutation in a rhodopsin gene may cause one very severe disease, which is called retinitis pigmentosa, which means progressive degeneration of the retina. One more role about the retinoic acid. As I said, retinoic acid is used as a hormone. Let's see, first students, every time you need to know about the transporters again. I said in general that they are transporters, specific transporters of all fat-soluble vitamins. It's only about the resorption, but after that, they can be saved in the liver, in the adipose tissue, and in the blood there are specific transporters. For example, for this retinoic acid, they are retinoic acid binding proteins, cellular binding proteins, and etc. Let's see the action of retinoic acid. After entering the cytosol, enters the nucleus directly because it is fat-soluble. Only fat-soluble compound can enter the nucleus and the cytosol without specific transporters because cell membranes are fat-soluble too. In the nucleus, they are recognized from specific parts of DNA, which is called a retinoic acid, a response element. But to be recognized of these elements in DNA, retinoic acid needs to be recognized from intracellular receptors, not membrane receptors, intracellular receptors, because they are lipid-soluble. They don't need any membrane receptors, but they need intracellular receptors. And these are two types of retinoic acid receptors. The first ones are called RAR, retinoic acid receptors means. The second ones are called RXR, which means retinoic X receptors. After the entry of retinoic acid inside the nucleus, there is a recognition with RAR and RXR and formation of heterodimer RAR plus RXR. Here, look at here, RAR, RXR. This heterodimer is recognized from a retinoic acid response element. And after that, starts activation of gene expression of something. Actually, this is the mechanism of action of all lipid-soluble hormones, not only about retinoic acid. D3. The first question is a vitamin D3 a vitamin? Vitamin or not vitamin? The answer is that vitamin D3 actually is vitamin because we can take from the food, but the action is as hormone. It is a hormone. And the name of this hormone is calcitriol. Calcitriol. This is the name of this hormone which comes from this synthesis. One more about D3. Actually, people know that vitamin D3 is synthesized in the skin. It's not correct in the skin because in the skin there is only the first reaction. Actually, the active form of vitamin D3 is formed in the kidneys, but not in the skin. Other myth about vitamin D3 is that sun is responsible for the synthesis of D3, or sun synthesizes vitamin D3. No, it's not correct. It is used only for one reaction, but not for full synthesis of vitamin D3. Let's follow the reactions. First, for the substrate of vitamin D3, we need one precursor, which is called 7-dehydrocholesterol. 7-dehydrocholesterol. This is the precursor. This is the substrate. Where comes from this precursor? Look here, students. This is the process of synthesis of cholesterol. This is the cholesterol synthesis. One of the compounds before cholesterol synthesis is called 7-dehydrocholesterol. This is the compound which is used for the synthesis of D3. It can be formed in the skin, yes, can be formed in the skin, this precursor. But can be transported from other organs, like liver, for example. From liver to the skin, can be, yes. After that, this compound, students, undergoes one very, very unique reaction. It's a reaction of photolysis. This reaction cannot be done from any of our human enzymes, cannot. We don't have any human enzymes that can disrupt, damage, broke any of the bonds in the structure of cholesterol. We don't have. Only sunlight, UVB rays, only these rays can be used for this. There is breaking of one bond between carbon number 9 and carbon number 10. That's all. This is the reaction from sun rays. And after that, there is some returning of the ring. It's not shown here. Yes, it's not shown here. And after that, this proform, which is called cholecalciferol, which is not active form, returns back to the liver. In the liver, there is enzyme 25-hydroxylase, and there is hydroxylation of 25th position. It is formed calcidiol, or 25-hydroxycholecalciferol. It's not fully active. Then this form goes to the kidneys, where there are two more enzymes. The first one is called 24-hydroxylase, and the product name is 24-25-dehydroxycholecalciferol. Some active, but not full. And And the last enzyme, which is key regulatory enzyme, the main enzyme in the synthesis of D3, is called 1-alpha hydroxylase. And the product is the hormone name, 1,25-dihydroxy D3, or calcitriol. Tri because there are 3 more hydroxyl groups. What is the action of D3? What is the biological role of D3? This is the classical biological role of D3, which is increasing the calcium in the blood. To increase calcium in the blood, students. How? There are 3 possible ways in general, never mind that we're talking about calcium or something else. There are 3 ways, logical ways, logic ways. The first way is, this is the main action of D3 actually, to increase the absorption of calcium in the small intestines. To increase the absorption of calcium in the small intestine. Here, in the apical surface. This is the action of D3. Why is possible and what does it mean increase the absorption? This means that calcium is responsible for the genetic expression of some protein transport channels to start the transport of calcium from the lumen to the blood. Understand? Why is possible? Because vitamin D3 is used as steroid hormone, can enter the nucleus, can be recognized from DNA, and can be induce the corresponding gene expression to activate the formation of these protein channels in the apical surface. Which means if you want to take calcium as supplement, need to be together with D3 to be absorbed, to be easily absorbed. The second action of D3 are bonds. Why bonds? Because to increase something in the blood, we can take it from some storage. The storage of calcium are bonds, that's why. And the third action are kidneys, to stop releasing bile-urine. This is the action of D3 in all these 3 places. But the most important is number 1, intestinal epithelium or enterocytes. Actually, students, there are 2 more hormones which are used for the calcium metabolism. The first one is called parathyroid hormone, which has the same action as D3, increases calcium in the blood. It is formed by parathyroid glands. And the third one is called calcitonin. D3, parathyroid hormone, and calcitonin is the third one, which is formed by thyroid glands. This is the formation of calcitonin. The action of calcitonin decreases calcium in the blood. Vitamin E, or tocopherols, include tocopherols and tocotrienols, if there is or not double bonds. It is used in this cycle together with vitamin C. It's absolutely natural antioxidant together with C. Both of the vitamins are powerful antioxidants in our body and can neutralize reactive oxygen species. And there is absolutely interaction with vitamin C. And let's start talking about enzyme classification, students. You have this information for today too. There are 6 main groups of enzymes, and let's start with the first one. The enzymes are called oxidoreductases. Actually, we talked about this reaction of succinate dehydrogenase enzyme. This is the substrate of the enzyme. This is the product of the enzyme. Here is the enzyme name under the arrow, and this is the cofactor of the enzyme name. To explain one reaction will be like that. Substrate is succinate, which is converted to fumarate, which is a product of the reaction. The enzyme name is succinate dehydrogenase, and the cofactor is FAD, which is converted to FADH. Actually, FAD is so-called co-substrate of the reaction. FADH is so-called co-product of the reaction. And in the end, we need to say if the reaction is reversible or not. It is reversible. The next reaction is about pyruvate and lactate. Lactate dehydrogenase enzyme, the same like in the board. Second types of enzymes are transferases. They are used for the transfer of some functional groups. The functional groups can be phosphate groups, methyl groups, carboxyl groups, no carboxyl groups. You see the carboxyl group transfer, it's a separate group. Can be sulfur groups, acetyl groups, and etc. There is one specific name for the transferases of phosphate groups. These enzymes that are used for the transfer of phosphate groups are called kinases. And this process, this type of the reaction is phosphorylation. When there is transfer of phosphate group, this enzyme reaction is called phosphorylation. Kinases are the enzymes that are used for the transfer of this phosphate group. Look, students, substrate is glucose, product is glucose 6-phosphate. There is transfer of only one phosphate group. This is the gamma phosphate group of ATP, which is taken from the ATP, is involved in the glucose on the 6th position. That's why the product is called glucose 6-phosphate. Enzyme name, hexokinase. Magnesium is used also in the reaction. Other example of transferase reaction is transfer of amino group. The enzymes are called transaminases. And what is the cofactor? What is this PLP? It is a pyridoxal phosphate, which was a derivative of which vitamin? B6. B6. Other types are hydrolases. They are used to with adding of 1 molecule of water across a bond, hydrolyzing it. The first example is carbon-sensitive lipase. This is the substrate, triacylglycerol, plus 3 molecules of water. The products are glycerol and 3 fatty acids. Glutaminase, another hydrolase enzyme, uses water again to hydrolyze this bond here, exactly here, and this amino group is released as ammonium ion, and the rest of the compound is glutamate. Number 4 reactions, lyases. Can you see the difference between this water and this water? This water is used to hydrolyze the bond. This is the role. But this water here is used as substrate, because this water is involved in the substrate. Substrate, fumarate. Product, malate. Enzyme, fumarase. One more substrate. The substrate is here. This is the hydroxyl group from water, and this is the hydrogen atom, which comes from the water too. One more example are decarboxylases. Isomerases, simple enzymes without any cofactors. The isomerases examples are this isomerase from glucose 6-phosphate to fructose 6-phosphate. This is the reaction from glycolysis. And this one is phosphoglucomutase reaction. This is a reversible reaction between glucose 1 and glucose 6-phosphate. It's a very important reaction. Remember reaction, students. Ligases are the 6th group of enzyme classification. There are big differences between lyases and ligases. Ligases are the 6th group of enzyme classification. Lyases are group number 4. Next example. Ligases use obligatory ATP. Lyases not. One more. Ligases are 2 types. The first types of ligases are carboxylases that use biotin. We said something about biotin together with ATP, but one more types of ligases are synthetases. There is difference between synthesis and synthetases. Synthetases means that they use ATP. Synthesis don't. Mechanism of enzyme catalyzed reaction means the formation of these intermediate complexes and in the end unchanged enzyme plus product. Active site, what is this? It's a part of the surface of the enzyme which composed of some groups, like catalytic group with red color, which take part in the reaction, contact groups that take part in the binding with the substrate, some additional and conformational groups there are in the active. All enzymes have active sites, no enzymes without active sites. Don't confuse active with allosteric. Active sites, the active sites are in all enzymes. This is the place of contact with the substrate. There are two enzyme specificity, students. The first is reaction specificity. You can see this example. If we have the same substrate, which is any alpha amino acid, with three different enzymes, because they are used to break different bonds, or they are typical for different reactions, different products will be produced. For example, amino acid oxidases. They are used for deamination and formation of alpha-keto acids. There is removing of amino group. Decarboxylases are used for the removing of carboxyl group, and the products are biogenic amines. And transaminases are used for the transfer of amino group between alpha amino acid and alpha-keto acid. Here, these are two substrates and two products. To summarize, different enzymes with the same substrate, different products. Enzyme specificity about the substrate. There are two types of substrate specificity. It can be absolute substrate specificity, like the enzyme urease for the urea, or arginase for the arginine, and not absolute specificity. Here the example is about the specificity to the group. If there is arginine and lysine, because of their groups, there is action of pepsin or trypsin, and etc. There are three models to explain the substrate specificity. The first model is a key-killer model, or the model Fisher, which means if there is substrate and enzyme, no changing of anything. They just are recognized and the enzyme substrate complex is formed immediately. No any change in both of them. The second model is called induced fit model, or model quotient. This model shows that if the substrate and the enzymes, both of them are like globular, after their contact, there's changing only of the enzyme. Enzyme is changed only. This is the second model. And the third model explains the changing in the both of them, in the substrate and in the enzyme. This is called induced fit model plus substrate tension. And that's all from today's students. Thank you for your presence today.